Today I will talk about a possible novel RNA-mediated transcriptional activation mechanism. I called it RNA memory, may be involved in cell identity. Okay, what is cell identity? Daughter cell is a concept by which, after mitosis, daughter cell will restore the same chromatin profile and the same transcriptional profile of the mother cell, with other feature, of course. Okay, what happens during mitosis? Usually in G0, G1, cells has a certain chromatin profile and a certain transcriptional profile, which functionally change during SG2 phase. Then, during mitosis, chromosomes are highly condensed, the picture of wrong, chromosomes are highly condensed and there is no transcription at all, almost. Then, by just partially understood mechanism, daughter cells are able to restore both the same chromatin profile and the same transcriptional profile. They are strictly correlated, they are linked, but they are not the same. Okay, this work was born on an hypothesis on a possible mechanism for keeping cell identity. Okay, actual models focus on explaining how is inherited an on-off gene status, not the exact transitional profile. Again, they are linked, they are not the same. In other words, when we talk about chromatin, we know that polyclone group proteins, trithorax group proteins, histone variants, DNA methylation, the big world of epigenetics, can explain how this phenomenon is inherited, these features are inherited. When we talk about the transcriptional profile of mother cell and daughter cells, there are just some hints about non-coding RNA which can interact in this phenomenon. The problem is that actual models don't explain anything. For example, it is seen that cell identity heritage is clonal. In other words, if there is a random variation in the transcriptional profile, daughter cell will inherit the variation of the transcriptional profile without a clear change at chromatin level. The classical example is the ratio between alpha and beta globin in erythroid cell lines. Usually the ratio is 1 to 1. When randomly, stochastically, some cells change this ratio, for example 1 to 2, all the cells will retain the same variation. Okay, so I thought there could be some missing piece of the puzzle, and therefore I formulate an hypothesis about that. It's not exclusively mine, there are other people who thought something like that, I add something. It's possible a cytoplasmic memory for cell identity via diffusible elements which could act in trans, such as RNAs. In other words, in the cytoplasm of the mother cell, there could be some remnants of messengers which mirrors its own transcriptional profile. After mitosis, these remnants could be read by daughter cells, and so the transitional profile could be restored in the daughter cells. There are some papers which could suggest a mechanism like that. For example, it's known in Drosophila that when PARP gene expression is impaired, the expression of PARP-E, which is an alternative splicing form for PARP which lacks its enzymatic activity, can restore both the presence and the activity, the enzymatic activity of PARP wild type. This is another paper, don't worry about that, there are other explanations for that. This is the most important paper we've found. This is an old paper of the 1997. Transient transfection of beta globin gene induced transcription at the chromosomal beta globin locus in non-erythroid cell lines. And this effect is not dependent on protein expression and therefore is RNA mediated. In this paper we see how a gene is able to induce transcription in its corresponding locus. And I avoided at all to talk about the implication of nuclear transplantation, nuclear transfer, transinduction, transvection, and so on. There are a lot of biological phenomena that could be explained with this model. To test my model, I choose to use Drosophila melanogaster as animal model and to test it using PV, position effect variegation. It's a very solid system. For everybody who don't know what the position effect variegation is, usually in Drosophila the white gene permits the product, permits the pigmentation of the eye. There are some mutations which brings this gene at the boundary between euchromatin and centromeric heterochromatin, and thus the white gene is randomly inactivated by the spreading of heterochromatin, giving this typical salt and pepper effect. Usually this phenomenon is used to have an insight at the global state of transcription of chromatin. Mutation which gives a redder high are mutations which achromatinize chromatin. They are called suppressor variegation or SUVA. Exactly the opposite for enhancer variegation mutation. You close the chromatin, you stop the transcription of the white gene, you have a whiter eye. I use this phenomenon just to have an insight on the transcriptional state of the white gene. I'm not interested in white. Okay, for me it's perfect because the white gene is not an essential gene. The white gene product is a marker of terminal differentiation. I cells randomly do not express it, but the cells are committed to express. There are not cross products. And PEV is clonally inherited in each materium from the first cell. Okay, I genetically introduce in the variegating flies some hypomorphic null, some phenotypically null or hypomorph allele for the white gene. If my model is correct, after mitosis, these hypomorph or null alleles, the RNA of these alleles, will be read as a sort of abundance of the white gene, for the production of the white gene. Thus, if my hypothesis is correct, I should have an euchromatinization of the closed white gene, of the heterochromatinized white gene. In other words, if my memory RNA hypothesis is correct, at last some white alleles must behave as suppressor variegation. From these flies, variegating flies, I will insert a white allele which cannot produce protein, and I will revert from this phenotype to this phenotype in vivo. Those are the stock I've used. I don't know if there's some genetics between you. Anyhow, these are the very enhanced stock. Here the white gene, the production of the white gene is almost impaired. They are almost white. This is the experimental design. On the left panel you see the progeny of the crossing between the variegating allele and the cray allele, both when the cray allele comes from the father or from the mother, because in Drosopia there could be a parental effect. On the right panel you see the controls. On the right up panel, you'll see the effect of the genetic background to see if there is some hidden SUVA. On the lower panel, you see the color peristalk background control. This is what I've obtained. As you can see, this is the most striking allele I found. That's the panel of alleles. If you can see, I obtained a strong suppression of variegation. Just adding. This is the allele when it comes from the father, this is when the cray allele comes from the mother. Here, striking. Just think, it's not exactly, but just think about this. I added these flies plus these flies, and I obtained this fly. I just reactivated this gene, absolutely. This is an independent experiment with different flies to avoid procedural errors, it's just another control. And think again, I've added, this is the background color of the allele. This is my variegating fly. I added these two flies and I obtained this kind of color. It's almost wild type. It's a striking suppression of variegation. So, what I obtained is that some null or hypomorph white allele can act as suppressor variegation, and this means that some null or hypomorph allele are able to induce in vivo the transcription of their corresponding gene. This work has been followed in Palermo, and a huge panel of white alleles have been tested. A lot of them respond to this test. A fine quantification of suppression has been performed using high-pigment assay. This is just to give numbers to the effect. It has been discovered it's not a classical PEV, it's gene-specific. And it's what I expect, because it must not be genome wide, it must be gene-specific. Again, it has been studied how this phenomenon is inherited. Just a quick comment. Some alleles act as a sort of activating paramutation. It's the first time it's seen. Some insight is arrested about the mechanism, but not so much. And there is a manuscript in preparation. Of course, there are a lot of things to do. Test other genes in Drosophila, find the conservative portion which gives this effect, a genetic screen to find interactors, creating transgenic flies with xenogene, and changing model, maybe human cultured cells, and a lot more. As conclusive remarks, I think that this effect will be mainly covered because it could need full-length RNAs and non-cDNA construct to be performed. There are some papers which could suggest the role of the first UTR in this phenomenon, raising interesting questions about the experimental drift, what I fear most, and the role of introns in the regulation of gene expression. Another, think about why in superior eukaryotes, usually nuclear membrane is disrupted during mitosis. It's not a must. Some eukaryotes can replicate without disrupting nuclear membrane. Maybe to allow cytoplasmic factor to merge with nuclear factors. Again, and I think this is what interests you most. Is it possible to postulate an RNA interference RNA memory system to fine-tune the transcriptional profile? Could this system be used to perform cellular programming? Could this be used in regenerative medicine?